I will get started. So the title of the talk today is single cell analysis of alternative splicing using a recently published software called Marvel. So what exactly is alternative splicing? So we have to always start with the genes. So a gene is essentially a series of exons, but how a gene use the different combination of exons would give rise to very different isoforms. So for example, if we look at this gene over here, when it uses the blue exon, it gives rise to isoform A. And when it uses the pink exon, it gives rise to isoform B. And quite naturally, different isoforms will give rise to different amino acids with different sequence, and finally giving rise to different protein structures. So I always love to use this apple to sort of to represent what alternative splicing is. So you can see that over here on the left-hand side and on the right-hand side, this picture of the same apple, but so representing the same gene. But you would see, if you take a closer look, you will see that the puzzle pieces that make up these two apples are in a slightly different configuration. So this is what alternative splicing is, using different combinations of exon from the same gene locus to give rise to very different protein structure and function. So why do we want to go beyond genes and study alternative splicing? Why shouldn't we? Why should we just stop at gene expression and not go further for splicing? So one reason is because different isoforms can give rise to very different protein functions. So for example, we know that different proteins arising from the same gene can sometimes mean the difference between good and poor survival. And sometimes it could also really mean the difference between a protein that is sensitive to a given drug or a protein that is resistant to a given drug. There are also many diseases out there that are associated with abnormal splicing mechanism. So for example, one of the ways that this splicing mechanism can go wrong is that we get a genetic mutation that affects one of the proteins that is involved in alternative splicing. Another way the splicing mechanism can go wrong is that we get an overexpression of certain RNA binding proteins. And the third way in which the splicing mechanism can go wrong is that we get a genetic mutation that affects the splice sites. So another reason why we should go beyond genes and study alternative splicing is because also when we do differential expression analysis, we probably will get certain genes that are differentially expressed. So over here, if we do, let's say, a differential expression analysis between two cell population or between two sample groups, you will get this really nice volcano plot where the genes that are upregulated are in red and the genes that are downregulated in blue. But after having your set of candidate genes, certainly you want to perform some kind of functional validation on this few selected genes. And so when the time comes to investigate this set of genes in more detail, I'm sure you would have at some point asked yourself which isoforms of these genes are actually expressed in your data set. And this is very relevant. For example, if you want to knock out or knock down a given gene, you will certainly have to design your single guide RNA for CRISPR or shRNA against exons that are actually expressed. You may also want to overexpress a given isoform for your genes, and this is particularly relevant for oncogenes, for example. Or you may just want to perform a simple qPCR validation. And when that time comes, you probably will be asking yourself which isoforms are expressed because you want to design PCR primers that are specific to exons that are actually expressed in your data set. So these are also very technical and very practical reasons for studying alternative splicing or isoforms on top of gene expression analysis. So why do we want to study alternative splicing at the single cell resolution? So alternative splicing at the single cell resolution is I would say, quite a new area of research, but there are actually good reasons for us to do this. So for example, if we look at the top panel over here, you will see this is the bulk RNA sequencing. And you can see that in terms of bulk RNA sequencing, the biological signals are actually kind of entangled. And the reason is simple. That's because at the bulk level, your sample is typically consisted of many different cell types. And so hence it's difficult to tell which genes are actually expressed by which cell type. But when you perform single cell RNA sequencing, such as the bottom panel below, you will see that the signals are actually disentangled and quite well delineated. So for example, over here, if you do single cell RNA sequencing, you will be able to understand which signal belongs to which cell type. And this is especially true in hematological malignancies, where we have the cancer stem cells sitting at the top, the progenitor cells, and these cells give rise to very dysfunctional blood cells. And being able to understand the signals, especially coming from the disease propagating cells, which are the stem and the progenitor cells, are really important, because these are the cells that gives rise to all the dysfunctional blood cells, and these are also the pool of cells that are probably very relevant for drug targets. So in conclusion, single cell is able to disentangle the source of the biological signals and attribute it to specific cell types that are very relevant for drug therapy. And so there comes Marvel. So the goal of Marvel is really simple. It's really to make alternative splicing analysis possible at the single cell resolution. And so Marvel is designed to analyze single cell RNA sequencing data that is derived from two main library preparation platforms. So on the left-hand side is a library preparation platform, which is plate-based, such as SmartSeq2. And the reason why it's called plate-based is because every single cell would need to be manually or semi-automatically processed using a 384 or 96 well plate. On the right-hand side is a RNA sequencing library prepared using a droplet-based platform. So droplet-based means that every single cell is processed in a very, very automatic fashion. And I believe the most popular platform today is the 10x Genomics. And so in my presentation today, I'm going to show you the proof of principle of Marvel for RNA sequencing data generated from both plate and droplet-based platforms. And we'll start with plate-based first, because plate-based arrived at the single-cell RNA scene before droplet-based did. Sorry, it shouldn't be Dropbox, it should be droplet. I must be thinking of Dropbox when I was preparing this presentation. Okay, so Marvel stands for multi-model approach to review single-cell splicing dynamics. And so like every software, it begins with the pre-processing. So I won't dive into the technical details on how to prepare the files over here, but I just really want to show you over here that Marvel is just like any other software that requires the sample metadata, gene metadata, expression matrix. But because we're studying splicing, we also need the splicing metadata. So once we have these input files, Marvel does everything automatically downstream. So the first part is really to identify the alternative splicing events that are actually supported by sufficient sequencing reads. So for example, over here, you will see that this is an alternative splicing event that is only supported by one read, whereas on the second panel over here, you will see a very confident alternative splicing event that is supported by many reads, suggesting is a true splicing event that is expressed in your data set. And so alternative splicing event at the exon level comes in 7 different flavors. So the first flavor over here is a skip exon that is sometimes included or excluded in your cells. The second splicing event is called mutually exclusive exons. So the reason why it's called mutually exclusive is because if the pink exon is expressed, the blue exon is not expressed. Conversely, when the blue exon is expressed, then the pink exon is probably not expressed. Retained introns means that the intron between the two exons is not spliced out. And then we have the alternative 5' and 3' splice site, where a portion of the exon is included. And finally, we have alternative first and last exon, where sometimes the isoform prefers to use a different starting exon, or the isoform may prefer to use a different last exon. So these are the 7 flavors of alternative exons or alternative splicing events that we would study in a plate-based RNA sequencing format.
One question can I ask? So for the splicing metadata, could you explain that? What is that files we need to use your tool?
Okay, so so Marvel is designed in a way that is agnostic to the softwares that are used to generate the splicing metadata. So for example, my favorite software to generate the splicing metadata is RMATS, RMATS. But you may use another splicing software to prepare the splicing metadata, and Marvel can still take in that. So so basically it's up to you.
Got it.
But I like RMATS, so if you can use RMATS, use that, but it's really up to you.
Thank you.
No worries. So once we have identified the alternative splicing events, we then have to, of course, quantify that value, isn't it? So for so how we quantify this is that we use a unit called percent splice in value, and it's actually a very simple formula. So over here, for example, if you look at this alternative splicing event, the percent splice in value is essentially calculated as the number of sequencing reads supporting the alternative exon divided by the total number of sequencing reads that are found to in that region. So in other words, the percent splice in value represents the percentage of isoform that expresses the pink alternative exon. So what does it mean in terms of a more a relevant example? So like if we have a PSI value of 100%, it means that in that cell, all isoform expresses the pink exon. If we have a PSI of 50% for that cell, it means that for that given isoform, only half of the isoform expresses the pink exon, whereas the other half of the isoforms do not express the pink isoform. And lastly, it goes without saying, if we have a PSI of 0 for that isoform, it means that that cell expresses the gene, but for the isoform, it doesn't include the pink exon. So finally, after we have done the very tedious pre-processing part, you're now really ready to perform some downstream analysis to review some biological insights. So like all softwares, we like to begin with clustering. So clustering here means that we use the splicing events detected to see whether we can use that information to differentiate the cells into different clusters on the PCA space, for example. And one thing nice about studying splicing patterns is that you can start to look into the different patterns of splicing, and this is really one of the advantage of studying splicing at the single cell. So over here, I'm going to go through, hopefully quite slowly, where we have 7 different splicing events, splicing patterns. And so we know that the PSI value, because it's a percentage, it can take any value between 0 to 100. So just a quick recap, 100 means that the exon is expressed, whereas 0 means the exon is excluded. So what do we mean by included splicing pattern? Included splicing pattern means that most of the cells would express the pink alternative exon. Excluded means that most of the cells would exclude the pink alternative exon. Bimodal means that we have two populations of cells. The first population of cells expresses the pink exon, whereas the second population of cells exclude the pink exon. Middle is a very interesting pattern. Middle means that in every cell, half the isoforms includes the pink exon, and half of the isoforms include the pink exon, whereas half of the isoforms exclude the pink exon. And finally, like everything that we do, we need an other category. So multimodal is that other category whereby the splicing patterns are all over the place. And so one thing nice about this splicing pattern is that for the splicing pattern, bimodal, middle, and multimodal is something that we can never ever see at the bulk level. And the reason is very simple. It's because at the bulk level, the average values would be 50%, because at the bulk level, the PSI represents the average PSI value across all the single cells. And so at the bulk level, you would get a PSI value of 50% for bimodal, middle, and multimodal, whereas at the single cell resolution, you are able to distinguish these three very different splicing patterns. Okay, so then comes to the cornerstone of every RNA sequencing analysis, which is the differential expression analysis. So one thing nice about Marvel is that we're able to integrate both differential splicing analysis and also differential gene expression analysis. And by integrating these two omics together, we are able to study the relationship between gene and splicing. So for example, over here, you will see the gene expression decreases from cell group A to B, and this is the same for the splicing pattern. For this opposing pattern, although the gene expression is higher in cell group B, you will see that the expression value is lower in cell group B. And finally, the third category is called isoform switching. So isoform switching is where there's no changes in gene expression values between two cell groups, but there is an obvious change in splicing pattern. So this is something that we can only pick up at the splicing level, but not at the gene expression level. Finally, to put things into context, we can perform pathway enrichment analysis on genes that are differentially spliced to understand whether these genes belong to certain biological pathways. And finally, we can perform nonsense-mediated decay prediction to understand whether alternative splicing gives rise to nonsense-mediated decay. So what this means is that when there's no alternative splicing, you will see that the transcript gets transcribed and translated to give the full-length amino acid sequence. But if there is abnormal alternative splicing, such as over here, where it introduces a new set of bases, and sometimes that disrupts the open reading frame by introducing a premature stop codon. And what happens to this transcript is that it doesn't get translated to the full-length amino acid, but it gets decayed into smaller peptides. So this is the one slide summary of what Marvel can do. And for the following slides, I'm going to show you the application of Marvel. So the first, so Marvel was initially, oh, Daniel has a question. If you want to ask right away, that's also okay.
For sure. The previous slide, when you say that you take as input the expression matrix, this expression matrix quantifies which feature, each one of the exons or the gene in total?
So the expression matrix is just your gene expression matrix. Marvel would quantify and generate the splicing expression matrix internally.
Okay, from the quantification of the gene itself.
From the quantification of the splice junction. Yes,
okay.
So it's a yes. So the splice junction matrix is one of the input files, but the gene expression matrix is also one of the input files.
I see. Thank you so much.
No worries. Okay, so so the story behind Marvel is that it was part of my PhD project. And so when I started building Marvel, it was meant for only plate-based RNA sequencing data set, because plate-based was the first kind of library prep to arrive at the scene. And so that's why I'm starting the story with Marvel on plate-based single-cell RNA sequencing data. And there's a few methods to generate plate-based single-cell RNA sequencing data. The most popular method is SmartSeq2, but there are also other methods available, such as SmartSeq3, or SmarterSeq, or TargetSeq. So choose your favorite platform. So for this proof of principle, I have chosen two very different cell types to show. The first cell type is induced pluripotent stem cells, and the second cell type is endoderm cells that has been differentiated from this set of stem cells. And so this is my favorite, this has always been my favorite figure to show for whenever I talk about alternative splicing. So the title of this is Splicing as an Additional Source of Complexity Under Gene Expression. So what this means is that if I use non-differentially expressed genes to try to separate these two cell populations, you will see that it doesn't really do a good job in separating these two cell populations. Whereas if I take the alternative splicing events from this set of non-differentially expressed genes, you will see that it successfully separates the two cell populations. So it means that when gene expression can't, splicing may be able to help you to separate your two cell population. And the reason is simple, because splicing may be actually an additional source of complexity underlying gene expression that you don't see at the gene expression level. The next is to do differential splicing analysis. So this is just one way of how I like to rank my differentially spliced exons, but you can also do it in your typical volcano plot, which I'm actually doing it these days. And so there were 1,616 differentially splicing events between these two cell populations. And so individual genes are nice to look at, but when we take this set of differentially spliced genes and do a pathway enrichment analysis, you will see some context. So the context here is that the differentially spliced genes are very much involved in the translation and transcription pathways, suggesting that these genes these cells are quite metabolically active compared to the stem cells. And we can also try to look into the relationship between gene expression and splicing. So for example, we can stratify the relationship between gene and splicing into three different categories. The first category is coordinated. It means that the direction of gene expression change is the same with the direction of splicing change. So for example, you will see that for this particular gene, the DHX9, its gene expression is downregulated in endoderm, and you can see that for this particular isoform, it is also downregulated in the same direction of the gene. Opposing means that the direction of gene expression change is in the opposite direction to splicing. So over here, you will see a downregulation of this gene in endoderm, but you will see that at the isoform level, it is upregulated in the endoderm population. And finally, isoform switching means that you don't see a change in gene expression, but in terms of splicing pattern, you will see a quite obvious change between the two cell population. So isoform switching is important because it's something that we cannot detect with gene expression, differential gene expression analysis, but we can pick it up when we perform differential splicing analysis. And lastly, we would also like to study the impact of alternative splicing. So one of the impact of alternative splicing is nonsense-mediated decay. And so what this means is that in the absence of alternative splicing, you would get the transcript that is translated to the full-length amino acid. Whereas if there is an alternative splicing event, such as an intron retention over here, there is a chance that this alternative splicing may introduce a premature stop codon. And so the consequence of this premature stop codon is that the amino acid that gets translated into is no longer full length, it's truncated. And so what happens to this kind of truncated amino acid is that you get subjected to nonsense mediate decay. So, so why is this important? So if we look at the gene expression profile for alternative splicing events that give rise to no nonsense mediated decay and those that give rise to nonsense mediated decay, you will see that the gene expression is decreased whenever there is an intron retention that leads to a nonsense mediated decay, as what we've seen over here. And so why is this important? So Marvel is able to overlay on the volcano plot for gene expression, the genes that are subjected to nonsense mediated decay, and which splicing event type this nonsense mediated decay is attributed to. So for example, you will see this gene over here, BUB3, which is very downregulated and is subjected to nonsense mediated decay due to the alternative 3' splice site splicing event. And so these genes that are subjected to nonsense mediated decay and at the same time downregulated may be potential candidate genes for your downstream functional studies.
Hey, Shin, we have a question on the chat.
Okay.
So Krishna asks if there is a correction in the analysis to understand the effect of read counts, because one of the issues of single-cell RNA-seq is that low number of RNA numbers can severely distort the splicing effects.
Yes, so so that would have been so splicing events that are lowly expressed would have been already been filtered out in the pre-processing step so that we can be confident with the splicing events that are actually included for the downstream analysis. And yes, if you're worried about okay, so if your next question is if the gene is expressed, but like you said, because of certain bottlenecks or amplification bias in the library preparation step, does Marvel take those biases into account? The answer is yes. So I didn't present it here, but in the paper that was published, we did adjust for this kind of inherent bias that is quite frequent in single-cell RNA sequencing data set. Okay, all right, so that, sorry.
Thank you for your answer.
No worries. So this is, so I've explained the proof of principle for plate-based. So as I was building Marvel, I realized that I couldn't just build a platform for RNA sequencing data generated from plate-based library preparation prep, because the whole entire research community is shifting so fast to droplet-based library preparation platforms. And so so I realized that I had to incorporate features to handle droplet-based library preparation RNA sequencing data set, such as 10x genomics. And so in the next few slides, I'm going to show you the application of Marvel on those data sets. So the first data set that I have chosen to show proof of principle is this data set where we have induced pluripotent stem cells that was differentiated into cardiomyocytes on different time points. So at day 2, day 4, and day 10. So this is a very simple volcano plot showing us that there are 575 junctions that are outregulated in cardiomyocytes day 10, and there are 243 splice junctions that are downregulated in day 10 relative to the stem cells. But what's really nice about this set of differentially spliced genes is that when we do a pathway enrichment analysis, we found muscle-related genes that are enriched among this set of differentially spliced genes, including pathways associated with muscles in general or energy production, for example. And so I always love to look into the gene splicing relationship, because by looking into the gene splicing relationship, we can really truly appreciate the reason for performing alternative splicing on top of gene expression analysis. So over here, we see the different categories of relationship, and we can see that more than half of this relationship falls under the category of isoform switching. It means that there's no change in gene expression profile between two cell populations, but there is an underlying change of splicing pattern between two cell populations. So as a proof of principle, we can see that there are two cell groups that I've chosen to do this analysis. The first is the stem cells, and the second is cardiomyocytes at day 10. And if we look at this gene called TPM2, you will see that there is virtually no there's virtually no difference in expression between these two cell groups. But if you look at the expression of splice junction number 1, you will see that its expression is more highly expressed in the cardiomyocytes. Whereas if you look at splice junction number 2, the pattern is the opposite. It is more highly expressed in the stem cells. So this illustrates the power of splicing analysis, where you can detect pattern that is invincible at the gene expression level. And what's nice about MARVEL is that it's able to perform a candidate gene analysis. So the previous slide looks into globally all genes that are expressed and to study them at the transcriptome wide level. What's nice about MARVEL is that we're able to choose our favorite gene and to plot some figures to give us more insights for that given gene. So for example, I've chosen TPM2 over here, and you can see that for TPM2, the expression progressively increases from the stem cell to day 2 cardiomyocytes and to later time points. If we look at the splice junction expression, you will see that for splice junction number 1, it is more highly expressed in the stem cells compared to the later time points. For splice junction number 2, it is expressed quite uniformly across all different time points, whereas for splice junction number 3, you would see that it is expressed in the later time points compared to the earlier time points. So this is just one example of a very complex splicing pattern where it shows quite different splicing profile compared to the gene expression. And lastly, MARVEL is able to plot the different isoforms just to show us where along the isoform the splice junction of interest is expressed. So for example, over here, if we look at splice junction 1, 2, and 3, which corresponds to this 3 splice junction, you will see that they are primarily expressed at the 3' end. Where splice junction number 1, which is more highly expressed, is located nearest to the 3' end, followed by splice junction number 2 and splice junction number 3. So this is just confirming what we know about droplet-based platform, where it's slightly more 3' bias. Next, I wanted to just show you that MARVEL can handle cells, many cells. So for example, over here, I'm showing you an analysis that we perform using more than 100,000 cells derived from brain tissues of individuals with autism versus individuals with no autism. So over here, because it's a because these cells were derived from brain tissues, you will see that there are many different cell types over here. And so if we, like every single cell RNA sequencing, we look into the differential splicing analysis first. And so over here, we identified 691 splice junctions that are upregulated in individuals with autism, whereas we identified 1,903 splice junctions, which are downregulated in individuals with autism. And so what's really nice is that if we look at the top most differentially expressed splice junction, it belongs to this gene called SYT1, which is a canonical marker gene for certain cell types in the brain. So because it's a canonical gene, you don't expect it to be differentially expressed between the controls and individuals with autism. So you can see that at the gene expression level, there's virtually no difference in its expression between the controls and the autism individuals. However, if we look at the splice junctions of this gene, you would see that it's more outregulated in the controls, and conversely, it is downregulated in individuals with autism. So then again, this example just highlights that there are some candidate genes that we just don't detect by performing differential gene expression alone, but we are able to detect when we perform differential splicing analysis. And lastly, when we look into the pathways that are involved for these genes that are differentially spliced, you will see that there's a lot of these pathways that are associated with the nervous system. So for example, pathways associated with the synapse, with the axon, with the dendritic cells, even tau proteins, which we know play a role in Alzheimer's, and RNA splicing itself, suggesting that we managed to identify biologically relevant genes that are differentially spliced. Yes, so I also like to just quickly talk about a accompanying software that you can use with Marvel. So Marvel does a good job in a global characterization of the splicing landscape between the different cell populations. But perhaps you might also want to, in some sense, visually validate some of your candidate genes just to make sure that they are actually spliced. So Valerie stands for Visualizing Alternative Splicing Events from Single-Cell RNA Sequencing Experiments. And this is just a one slide to show you its proof of principle. So to show you the benefits of Valerie, I've looked into this data set consisting of stem cells differentiating into motor neuron cells and neuroprogenitor cells. And the alternative splicing event of interest here is the mutually exclusive exons of PKM. So mutually exclusive means that when one exon is expressed, the other is not. So if you were to use a conventional genome browser to look into this splicing event, you would see, for example, for sample number 1, you would feel that exon number 9 is more highly expressed. But if you look at sample number 2, exon 10 is more highly expressed, suggesting that there's some underlying heterogeneity that we cannot validate when we look into the bulk level and look into the IGV or genome browser, for example. You may want to choose some single cells to validate on the genome browser. But then again, the same issue arises. If you look at sample 1, 2, and 3, cells 1, 2, and 3, you will see that exon 10 is more highly expressed, but exon 9 is more highly expressed in sample number 4. Whereas for cell number 5, there's totally no expression at all, suggesting that even choosing just a few single cells to look at on the genome browser may not give you something that is representative of your data set. By looking into Valerie, Valerie is able to show you the broad picture of all cells that were included for analysis. So over here in this heat map, you will see that the rows corresponds to the different cells stratified by the different cell groups, red, green, and blue. The column represents the individual genomic bases, and the colors represents whether the exon is expressed or not. So you can see broadly for cell group, that for the red cell group, exon 9 and exon 10 are expressed in this cell group. The same for cell group in green, whereas for the blue cell group, exon 10 is not expressed at all. Exon 9 is not expressed at all, but exon 10 is. When we collapse this signal using the average, you can see more clearly that for exon 9, it is most highly expressed in the red cell group, followed by the green and the blue, whereas for exon 10, it is more highly expressed in the blue, followed by the green, and followed by the red. And to put a p-value to this, you will see that the exon expression is very different, statistically different, at the genomic basis corresponding to exon 9 and 10, which is the mutually exclusive exons, but it is not different at the black exons, which are the constitutive exons as expected. So finally, once you have gone through the pipeline for Marvel-Valerie, the whole idea is so that you would have a set of candidate genes that you can, genes or isoforms that you can functionally validate in an in vitro or in vivo model. So what's more to do for Marvel? There's actually still a bit more work that I would like to work on. And if you feel that you have the expertise to contribute, please don't hesitate to reach out. So why do we want to do more work for Marvel? So the goal is very simple. We want to go from 100 to more than 1,000 splice genes that we detect from differential splicing analysis to perhaps 1 or 2 splice genes that is actually feasible to validate in the experimental setting. So how to do this? One way to choose genes is that we can choose splicing events that has an impact on the gene. So one feature that I've already shown you is alternative splicing can lead to nonsense mediate decay. And sometimes this leads to a gene that is no longer, a protein that is no longer functional. And this is quite relevant, for example, for tumor suppressors. The other side of the coin is that the alternative splicing do not introduce the premature stop codon. And so what happens is that we get a protein that is potentially hyperactivated. And this is very relevant for oncogenes, for example. Another function that we can think of adding to Marvel is to, for example, to see whether the certain alternative exons are enriched for certain sequence motif. So sequence motif can often give hint to the kind of splicing factor or RNA binding protein that binds to that given exon. So that can give you a hint for mechanism, for example. So these are things that I'm working on at the moment. And if you'd like to lend a hand, don't hesitate to reach out. So yes, so the goal is really just to get from many candidate genes at the beginning to filter down based on functional annotation to a set of smaller genes or isoforms that we can validate in the lab. So Marvel and Valerie are both publicly available softwares for non-commercial entities. And so it's available through two platforms. The first is GitHub, where the most up-to-date version is with new features and with tutorials. So I suggest always go to GitHub for the latest stuff. The ones on CRAN are the versions that was published according to the paper, and the up-to-date or the new features will come more slowly for the CRAN version. So I've shown you the application on certain cell types and certain circumstances because those are published data set. But my interest really lies in cancer and hematology in general. But I can't show you the application on these two areas of research because it has not been published yet. But perhaps I will have the opportunity next time. And
okay. So we have one, yeah, I think that you are going to close this talk. We actually have one question. Ashish, do you want to finish this slide before we? I'm not sure. Shine. How do you want to do that? Do you want to close this talk and then answer the question, or you can answer the question?
Yes, I think I'm quite happy to close this talk because it's quite done. But I think it's just nice to acknowledge the different individuals who has been very supportive to complete this Marvel journey. So I'm happy to take questions if any. So
there are two questions right now. One is from Ashish. Ashish, you can talk now.
Yeah. Hi. So Dr. John, so like in your last slide, you were talking about various things that can happen to proteins. So have you also considered chances when the protein fold, there is problem with that, and then that can lead to the protein degradation? So maybe that you can also talk about.
Absolutely. So so you're absolutely right. When the exon is included in the transcript and it gets translated into the protein, it may not always lead to hyperactivation, but like what you said, it could potentially lead to misfolding. So this is a predictive function that we can take into account when we add this to MARVA.
Yeah, thank you.
Great. We have two more questions. One is from Biju, the other one from Alice. Let's do that one by one. So Biju, you can ask the question first.
Yeah. Hi, Dr. Sean. Thanks again for the marvelous talk. I was impressed. So my questions are really technical. One is regarding the 10x data that you're processing, right? So how does MARVA, given the technology, 10x technology uses the 3' or 5' libraries, quantify exons that may be in the middle and maybe there is no coverage, unlike a SmartSeq2 technology, right? And then second question would be, it'd be really a future, probably a question for MARVA. Does MARVA consider a gene fusion event, to quantify, or is there a plan to do something like that?
Okay, so you're absolutely right. So that's one of the problems that we faced when we were looking into 10x genomics, where the data is quite sparse. So basically, if we look at this slide over here, we quantify the splicing events at the splice junction level instead of the exon level, because splice junctions are just more feasible compared to exons when we analyze droplet-based data. And the second point, you're absolutely right. Because of the inherent bias in droplet-based, you can see that we are only powered to analyze splice junctions that are towards the 3' end over here. So you can see that the most highly expressed junctions for this gene, TPM2, is splice junction 1, 2, and 3. And you can see that coincidentally, they're all located at the 3' end over here. And there may be some genes that are more highly expressed, and probably some splice junctions were detected in the middle. And if they were detected, we will analyze. If it's not detected, then we just can't analyze it. And that's just the limitation of the library prep system.
Okay, thank you. Great. And regarding my question on gene fusion, is that something that you would consider at some point?
So gene fusion is, I think it falls under the category of structural variation. And so structural variation is always very, very tricky. And so I would probably consider structural variation probably not strictly a splicing event, although it is, because you have two genes fused together and you get some abnormal splicing, but I think there's no immediate plans for that at the moment.
Okay, thank you.
Okay, we have one more question from Alice. I'm not sure what Alice wants to ask this question, or basically the question is in the Q&A, so I can read that.
Yeah, so actually, basically, my question is almost have been addressed in the previous question. So because when you show us some number of reads supporting each junction, I thought the count was pretty high. So I was wondering whether it was 10x data and your data was particularly more deeply sequenced than usual. And how fruitful you think it would be in terms of splice analysis using droplet-based method. Maybe you can also comment on the single cell-based approach versus some kind of pseudo bulk type of analysis to improve sensitivity with junction detection from 10x data.
Absolutely. So you're right that for each cell, the read counts are very low, or for some cells, no counts at all. So when we do the differential splicing analysis, we actually do it at the pseudo bulk level.
I see. Okay, that makes sense. Thanks.
Okay, so you basically do a pseudo bulk for each cell type. Is that right?
Yes, that's right. Yes.
Okay. All right. Well, I don't think we have any other question, but I do have one or two questions. So the first question I have is maybe this is a general question. So so can you tell us, like, when should we actually consider like single cell RNA splicing analysis? Usually we do the bulk, like for example, bulk RNA-seq or single cell RNA-seq, the kind of general analysis, identify the cell types and then do the differential expression analysis. Could you actually kind of give us some guidance, like in which case we should use, what we should do is like do the single cell splicing analysis?
Okay, so well, at the end of the day, gene expression is an aggregate of isoform expression. And so I think we should always begin our single cell analysis with gene expression. And then once you finish your gene expression analysis, you might be interested to understand which isoforms are actually driving the changes that you see at the gene expression level. Or you may also want to identify isoforms that is active in your data set, but you don't see at the gene expression level, so that you have more candidate genes to move forward with. And also, when the time comes to perform functional validation in the laboratory, your colleague is going to ask you which isoform is expressed, because they need to build the construct or they need to design the single guide RNA to specific exons of the gene. And so that's when splicing analysis will be helpful, because it helps you answer the question, which isoforms are most relevant for your gene of interest.
Yeah, so most of the time, for my experiences, like we do the bulk RNA-seq or single cell RNA-seq until we cannot find any differential expressed gene, or we didn't find any interested, differential expressed gene. Then we actually switch to the RNA splicing analysis, see whether we can find any differential and spliced transcripts. So that's most of the case. I also was curious, like in what kind of case we actually try to do the single cell splicing analysis. But I think it's yeah, I think that sounds reasonable, your explanation. The other question I have is that we actually use several RNA splicing software like RMAT and leafcutter and DXseq also. So for your excuse me. So for your current tools, you said that basically we can use output from those or any data format for like RMAT, and also any other softwares format we can also use in your software.
Oh, okay. So I'm just going to touch a little bit on your first question first, where you say when to do single cell, isn't it, right? Because sometimes you do your analysis at the bulk, and that may not be very insightful. So it's only helpful to do single cells when your original bulk sample consists of many different cell types. So for example, if you have sample from the, let's say, bone marrow or brain tissue, obviously that tissue is going to consist of many different cell types. And if you do analysis at the bulk level, you will not know your signal is coming from which cell type. So that's where single cell is very helpful, because it can tell you the signal is coming from which cell type. But let's say if you perform an experiment in a homogeneous cell line, for example, then the bulk level will be sufficient, because every cell in that sample is the same. So and single cell won't provide any additional insights, if that makes sense. And the second, just to answer your second question, yes, it can take in the input, the splicing events detected from different software such as RMAT and leafcutter. All you need to do is just to format it in the right way, and it can do the rest for you. I'm also using Marvel for bulk analysis, because in principle it can. It's just that you can do less things without. You can do less things without, but you still can use Marvel for bulk analysis as well if you want to. Yeah.
Got it. Great. Well, maybe I have one more question. So when you actually do this, when you do the RNA splicing analysis in the single cell RNA-seq data, so you still use the well-known markers to basically use the gene level data to do the annotation, right? The cell type annotation. You don't use any kind of differential spliced transcript to do the cell type annotation. Is that correct?
Yes. Yes, we still use gene expression for the annotation and to generate the UMAPs, TSNE, PCA. Got it.
All right. We're very interesting talk and great talk. Thank you so much for the presentation and also what are nice answers. I don't think we have any more questions from the audience. Great. Do you have anything else to say, Shine? If not, thank you so much.
Yes, thank you for listening, and I hope it has been useful to some extent.
Definitely. All right. Have a nice day. Thank you, everyone.